Namaste students, my name is JVN's Dr. Neeraj Dhulia, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapeeth, Women's University, Jaipur. Today we are going to discuss a topic that is culture techniques in diagnosis of disease. So this belongs to the subject medical biotechnology. So what are the culture techniques and why they are used and how they are used? So the culture techniques is generally utilized when the disease is not accurately diagnosed, or if it is diagnosed, it is not treated properly. Say for example, the infection is there, the doctor know that it is a bacterial infection and it is giving the antibiotics, but the disease is not cured. So it may be the possibility that the bacteria, the proper bacteria is not diagnosed by the doctor. So it may misunderstood by the doctor that some other bacteria is infecting and it is the antibiotic is not working on that. Or it may be possible that the bacteria is the same, but it have the antibiotic resistant bacteria. So for know that the which of the antibiotic will work properly, so we grow the bacteria on the artificial the supplied culture medium, and on that we put the antibiotics. So we can know that this bacteria is sensitive towards these types of the antibiotics and it is resistant to these antibiotics. So if the bacteria is resistant for say the A antibiotics, so the doctor will not prescribe the A antibiotic for that patient. Okay, so the alternative antibiotics and the antibiotics towards the specific bacteria is sensitive, that antibiotics will be prescribed by the doctor. And in some cases, the exact diagnosis is not observed, or it is not assumed by the doctor. So for the diagnosis purpose, the culture techniques are also used. So for example, which bacteria is infecting and which fungus is responsible for the disease and which virus is there. Okay, so the infectious diseases are often diagnosed by the culturing the samples isolated or harvested from the patient. So this is very much required for the exact treatment. Okay, so the lab technicians will take the sample and they will grow the bacteria, virus, or the fungus on the different types of the media. Okay, the selective media are there, the inhibitors that can be used. It is possible by the culture techniques to know the exact identification of the bacteria or virus or fungus and the sensitivity also. So what is the exactly the growing culture medium? Okay, so the microbes are there, the bacteria is there, fungi is there, the parasites are there, viruses are there. So they have the choices of their proper growth. Okay, some of the specialized media is there, the microbe will prefer to grow on them. Okay, so we have the studies that which type of media or which type of the specific factors are there for the specific types of the microbes. So we provide that specific type of the media and the bacteria, virus, or the funguses will grow, and we recognize by observing their appearance, colony appearance, by observing their in them into the microscope and by other means. Okay, so the precise characteristics of the growing culture can be used to identify a specific microbe, and use of the selective agent can be used to determine the features of the microscope microbes. Okay, so for example, the common bacteria which is causing the sore throat or the infection in the throat is the Staphylococcus aureus. and it may be resistant to antibiotics. Okay, so the doctor is prescribing the antibiotics, but it is not working because this bacteria is resistant towards that specific antibiotic which is doctor is prescribing. Okay, so we have to check the sensitivity of that bacteria towards the antibiotics. So these cultures are usually they contain the different types of the nutrients that the microbes like to grow, and this allows the lab technicians to identify that which microbe is growing. Lab technicians may need to grow the microbe on multiple different culture plates. Okay, so for say example, the 5 different culture media are there, and we have inoculating a single sample in 5 different types of the media to know that which bacteria is that, which microbe is that. So pinpoint exactly the which microbe it is. Okay, so the various types of the microbes can be diagnosed by the culture method, and the samples may vary according to the disease. Okay, so if it have the urinary tract infection, so the sample will be urine sample. Okay, if it is causing the diarrhea or another intestinal disease, then stool may be the sample. Okay, if it is associated with the STDs, then genital tract will the fluid from the genital tract or sample from the genital tract will be the sample. If it is causing the sore throat, then the swab will be the sample. If it is causing the other infection, then that specific site, that specific organ, that specific the fluid which is secreted from that specific part is or will be the sample. So the samples isolated from other body parts such as blood and other fluids may also be works as a sample. Okay, so various types of the techniques are there, a variety of alterations are there. Mainly the 3 types of the culture are there. One is the solid culture, then liquid culture, and on the culture on the cells or cell lines. Okay, so what is the solid culture, what is liquid culture, and what is the culture on the cell lines? So we will study one by one. So first is the solid culture. So the bacteria, most of the bacteria and funguses can be grown on a solid surface or solid culture. So the solid culture is it is a composition or mixture of the nutrients, various types of the nutrients, which enhances the growth of the bacteria and funguses. As a solidifying agent, we can use the gelating agent, that is the agar powder, and that is isolated from a sieve. So this is often on a plate, a culture plate may be used, or a test tube can also be used for the solid cultures. So in that the culture media, the composition of the media may vary according to the species. Okay, and the colors may be different according to the composition. It may be red, some may be yellow, or other colored media. So a single microbe placed on a solid surface can grow into colonies, and colonies are the clones of that particular bacteria or fungus. Okay, so we can observe the colony morphology. Okay, colonies are made up of the clones, and in which the cells are identical to each other. So the feature is what makes solid culture so useful for maybe microbial identification. Okay, so you can just observe the color of the colony. You can check the size of the colony, shape of the colony, the transparency, the corners of the colony, the shape of the colony. Okay, so that is the identification marker for the specific type of the bacteria and fungus. Then liquid culture are also there. Okay, so in few cases liquid cultures are required. So a liquid culture is grown in the media or a broth of nutrients. So microbial growth is observed for how quickly the broth becomes cloudy. Okay, so the time is measured by or associated by the number of the initial microbial density in the initial sample. So in this type of the culture, because you cannot identify the different colonies, so it gives the growth of the mixed colony. Okay, so this is not very much useful for the identification. Okay, so it may be useful for other types of the infection, for say for example the infection of the parasites. Okay, so that generally do not grow in the form of the colonies. Okay, so that is the use of the liquid cultures. Then cell culture, what is the cell culture and why it is used in the diagnosis? So some of the microbes, for say example, the viruses, they are not possible to grow on the artificial medium. So they need the animal cells, they need the human cells to grow. Some viruses need the plant cells to grow. Okay, so they need the live cells. Okay, for example, the chlamydia, the rickettsia, the viruses and viruses cannot be grown on the solid and liquid cultures, only cultures. So we have to provide the cell lines to them, human cells, animal cells, and the viruses infects the cells and they grow in the cells. So we observe and this is required to identify the exact viruses. What is the size, shape of the virus, which characteristics it is showing. So that for that purpose we need to grow the microbes, we need to grow the viruses. Okay, so the composition varies according to media, but some basic components should be there. Say for example, the nitrogen source is in the form of the digestive proteins. The growth factor should be there, so we can provide in the form of the yeast extract, serum, blood. The energy source should be there. So for example, the sugars, the carbohydrates may be added. One buffering agent should be there for the maintaining the pH, that the pH, phosphate can be used, citrate can be used. Then minerals are required. Okay, the selective agent for knowing the sensitivity. So chemicals, the antibiotics, some inhibitors are required. Okay, indicators are required. And if it is a solid media, then gelling agent is required. So this was all about the culture methods in the diagnosis, and hope you are satisfied with this detailed session. If you have any doubt, any query, you can write in the comment box. And this session is powered by Digital Version 2, Jyoti Vidyapeeth, Women's University, Jaipur. Thank you.